students, and welcome to the lecture on biocatalysts from extreme thermophilic and hyperthermophilic archaea and bacteria. The main objectives of today's lecture are to give a general idea about thermophiles, a brief discussion about classification of thermophiles, to discuss thermozymes and major applications of thermozymes, to explain in brief about biocatalysis, to give brief account about survival mechanism of thermophiles. To begin with, the first part of today's lecture, that is, to give a general idea about thermophiles. Thermophiles are organisms that live at relatively high temperatures of at least 60 degrees. Generally, microorganisms with an optimal growth temperature between 60 and 80 degrees are designated as thermophiles, whereas those growing optimally at temperatures of more than 80 degrees are referred to as hyperthermophiles, which are found in the three domains of life, that is archaea, bacteria and eukarya. But the majority are archaea and bacteria. Enzymes from thermophiles and hyperthermophiles are thermostable and display irreversible protein denaturation only at high temperatures. Thermophilic enzymes can be used in several industrial processes in which they can replace mesophilic enzymes or chemicals. The biocatalysts called thermozymes produced by these microorganisms are proteins that function under high temperature conditions. Due to their extreme stability, thermozymes offer new opportunities for biocatalysts and biotransformation. Examples of thermozymes include cellulases, amylases, xylanases, proteases, pectinases, keratinases, lipases, esterases, catalases, peroxidases and phytases, which have great potential for application in various biotechnological processes. Thermozymes can be applied as biocatalysts in existing industrial processes to replace presently used, often polluting chemical reagents. This is especially true for the pulp and paper industry, which has problems with aggressive waste from the bleaching process. Moreover, reactions carried out at high temperatures can be advantageous in cases in which the substrate or the product has poor solubility at low temperatures and or the thermodynamics of the reaction causes higher yield at higher temperatures. Genomic studies of thermophiles have demonstrated that the evolution of thermal tolerance depends on the levels of heritable variations such as genomic size shrinkage, horizontal DNA transfer and gene mutation. The omics data of thermophiles delivers a clear and overall view of the macromolecules participating in heat shock responses. Now, we will discuss the second objective of our topic, that is classification of thermophiles. Thermophiles can be generally classified into moderate thermophiles, extreme thermophiles and hyperthermophiles. Extreme thermophiles growing optimally at 60 to 80 degrees are widely distributed among the genera Bacillus, Clostridium, Thermoanerobacter, Thermus, Fervidobacterium, Rhodothermus. On the other hand, most of the hyperthermophiles belong to the archaea, which consists of four phyla: Synarchaeota, Euryarchaeota, Corarchaeota and Nanoarchaeota. Some genera belonging to the Synarchaeota are Sulfolobulus, Acidineus, Pyrodictium, Pyrolobus, Pyrobaculum, Thermoproteus and Thermophilum. Thermophiles can also be classified as follows. Number 1. Obligate thermophiles, also called extreme thermophiles, require such high temperatures for growth. And number 2. Facultative thermophiles, also called moderate thermophiles, that can thrive at high temperatures but also at lower temperatures below 50 degrees. Hyperthermophiles are particularly extreme thermophiles for which the optimal temperatures are above 80 degrees. Many of the hyperthermophiles archaea require elemental sulfur for growth. Some are anaerobes that use the sulfur instead of oxygen as an electron acceptor during cellular respiration. Some are lithotrophs that oxidize sulfur to create sulfuric acid as an energy source, thus requiring the microorganism to be adapted to very low pH, that is, an acidophile as well as thermophile. These organisms are inhabitants of hot, sulfur-rich environments usually associated with volcanism such as hot springs, geysers and fumaroles. In these places, especially in Yellowstone National Park, zonation of microorganisms according to their temperature optima occurs. Often, these organisms are colored due to the presence of photosynthetic pigments. Dear students, let's talk about next objective, that's thermozymes and the application of thermozymes. Cellular components of thermophilic organisms are also thermostable. Apart from high temperature, they are also known to withstand denaturants of extremely acidic and alkaline conditions. Thermostable enzymes are highly specific and thus have considerable potential for many industrial applications. The use of such enzymes in maximizing reactions accomplished in the food and paper industry, detergents, drugs, toxic waste removal, and drilling for oil is being studied extensively. The enzymes can be produced from the thermophiles through either optimized fermentation of the microorganisms or cloning of fast-growing mesophiles by recombinant DNA technology. The industrial needs for such specific thermostable enzyme and improvements required to maximize their application in the future are also suggested. Since the discovery of thermophiles in 1970s by Brock, the study of thermophilic and hyperthermophilic microorganisms has become a major domain of research. Increasingly, attention is being paid to these microorganisms because of their unusual properties. Their enzymes have unique characteristics such as temperature, chemical, and pH stability. Thermozymes are being used more frequently in several industrial processes. Enzymes from thermophiles and hyperthermophiles are thermostable and display irreversible protein denaturation only at high temperatures. Extreme thermozymes are enzymes that have their maximum activity above 60 degrees and hyperthermophilic enzymes have their maximum activity above 80 degrees. Thermophilic enzymes can be used in several industrial processes in which they can replace mesophilic enzymes or chemicals. The enzymes, particularly those that are active and stable at temperatures up to 100 degrees, are used to study protein adaptation to high temperatures and to obtain a better understanding of protein stability in general. Now, let us discuss about the application of thermozymes. Recent developments clearly show that thermophiles are a good source of novel catalysts that attract great industrial interest. Thermostable polymer degrading enzymes like proteases, pyrolynases, amylases, xylanases, and cellulases are expected to play increasingly important role in food, chemical, pharmaceutical, paper, pulp, or waste treatment industries. Now, let us discuss them one by one. Number 1, proteases. Proteases hydrolyze proteins into amino acids and peptides. Thermostable proteases are already used in several industrial processes. In most cases, they are used for their stability at higher pHs or in organic solvents rather than their stability at elevated temperatures. The best characterized thermostable protease is thermolysin, produced by Bacillus thermoproteolyticus. The use of proteases at high temperatures give rise to a specific problem. Heat enhances autolysis, that is, self-digestion of proteases. Their salting conformational unfolding leads to much faster autolysis because the partially or unfolded form of the enzyme is particularly good substrate for the protease molecules that are still active. The better susceptibility of the substrate to proteolytic attack results in higher specific activities for the proteases from thermophilic origin. Because most commercial substrates for proteases are mesophilic proteins, proteases have a high industrial potential. A method to prevent autolysis might be immobilization of the enzyme. Another actively marketed thermostable protease is thermus protease, used to clean up DNA before amplification in polymerase chain reaction. A larger application might be the use of thermophilic enzymes in domestic dishwashing detergent. In this case, much lower pHs are applied, combined with temperatures of about 60 degrees. Second is the alpha amylases. Alpha amylases are endo-acting enzymes that cleave alpha-1,4 bonds. They are used in the first stage of starch conversion, that is liquefaction. Raw starch consists of insoluble semicrystalline granules on which most enzymes are poorly active. The first step consists of heating the granules in water, which causes swelling and gelatinization. Then starch is liquefied by the thermostable alpha amylase from Bacillus licheniformis or Bacillus amyloliquefacens. Various enzymes can be used depending on the desired product. The most thermostable alpha amylase to date is from Pyrococcus vosei. It remained active after autoclaving for 4 hours at 120 degrees. Moving on to the next, that is glucoamylases. Glucoamylases are exo-acting enzymes that cleave both alpha-1,4 and alpha-1,6 bonds. These enzymes are currently used in the second step of starch conversion, in which they are used for the production of high glucose syrup from starch-originated polysaccharides. For industrial production, the glucoamylases from Aspergillus niger are generally used. Because of the lack of commercially available thermostable enzymes, the process conditions after starch liquefaction must be changed. For the production of glucose, the pH and temperature must be lowered. The next we have pyrolynases. Type 1 pyrolinase cleaves alpha-1,6 bonds in starch and is an example of debranching enzyme. It's used in the production of high glucose syrup from polysaccharides. For industrial purposes, the pyrolinase from Bacillus acidopyrolyticus is often used in combination with the glucoamylase from Aspergillus niger. Thermostable type 1 pyrolinase from Thermus caldophilus and Fervidobacterium panavorens has been characterized, and the latter has recently been cloned into E. coli. In addition to alpha-1,6 bonds, type 2 pyrolinase cleaves alpha-1,4 bonds and is therefore able to sacrifice starch directly into maltoesterose and maltotriose and maltobiose. Type 2 pyrolinase is usually referred to as amylopyrolinase. Several thermostable type 2 pyrolynases have already been described and purified. 2 hyperthermophilic type 2 pyrolynases from P. vosei and Pyrococcus furiosus have been expressed in E. coli. Next is beta-galactosidases. Galactosidases hydrolyze sugar bonds. The best known hyperthermophilic beta-galactosidases are from Pyrococcus furiosus. Most industrial applications of beta-galactosidases have employed thermostable glycosidases for hydrolytic purposes. For example, beta-galactosidases used in the hydrolysis of lactose into glucose and galactose. The monosaccharides that are formed from this reaction have higher solubility and sweetening effect than lactose and are more easily digested by humans. The main reason for using thermozymes in this process are increased substrate and product solubility and reduced risk of microbial growth. Glucosidases are also used in the reverse reaction of oligosaccharide synthesis. The thermostable Pyrococcus furiosus beta-galactosidase produces tri- and tetrasaccharides with lactose as a substrate. These oligosaccharides may be used as prebiotics in functional foods. Next is xylanases. Xylanases catalyze the hydrolysis of xylan. Xylan is the main polymeric compound of hemicellulose. Xylonases from thermophiles can be used in the biobleaching of wood or bagasse pulp. This is of great interest because the use of chlorine chemicals can be reduced, thereby minimizing environmental concerns. Minimal attack on the cellulose fiber is a prerequisite, and for this reason the xylonases should be cellulose-free. During the enzymatic bleaching process, the polysaccharide chains attached to the lignin hydrolyze and the pulp structure opens up. In this way, the consumption of bleaching chemicals can be reduced, while as higher fiber optical brightness is obtained. Commercial enzymes such as pulpzyma and cartazyme are not thermostable. In the treatment of pulp, several stages before the chlorine bleaching step are performed at 70 degree and higher. Consequently, pulp must be cooled before the enzymatic treatment and reheated for subsequent processing steps. In addition to obtaining improved thermostability, it is also desirable to have enzymes that are active at the alkaline pH of the pulp. Next, we will discuss about DNA processing enzymes. First is DNA polymerases. DNA polymerases are the key enzymes in the replication of DNA. Currently, thermostable polymerases play a major role in several molecular biological applications such as DNA amplification, sequencing, and labeling. A major advance in molecular biology is the development of PCR, that is polymerase chain reaction. In the first polymerase chain reaction, a heat labile E. coli DNA polymerase was used, which had to be added during each cycle following the denaturation and primer hybridization steps. The availability of thermostable DNA polymerases enormously facilitated the automation of the thermal cycling. The polymerase from Thermus aquaticus, Taq polymerase, is the first characterized thermophilic polymerase and is still widely applied in polymerase chain reaction. Thermostable DNA polymerases can also be used for reverse transcription, in which RNA is used as a starting template by first converting RNA to cDNA. This can then be used to generate cDNA libraries. With the original mesophilic reverse transcriptase that was used at lower temperatures, there was the problem of formation of stable secondary RNA structures. However, many thermostable polymerases can also use RNA as a template. The DNA polymerase from Thermus thermophilus is commonly used for this reaction. Next is ligases. DNA ligase catalyzes the linking of polynucleotides. The first thermostable ligase was discovered in Thermus thermophilus in 1984. Several thermostable DNA ligases have been discovered since then. An important application for thermophilic ligases is the ligase chain reaction. In this reaction, DNA ligase amplifies DNA, but a single base substitution prevents ligation and is thus distinguished. Therefore, it can be used in DNA diagnostics for the detection of single base mismatches. The specificity of this reaction is highest at the melting point of the primers, which is typically 50 to 65 degree. Another application lies in the construction of sequencing primers by high temperature ligation of hexameric primers. Then we have lipases. Lipases hydrolyze tri-, di- and monoglycerides into fatty acids and glycerol. Several hyperthermophilic lipases have been isolated and characterized. Biological treatment of fats at high temperatures is advantageous, since fats above their melting point in the liquid state become more accessible to the enzyme. Although oil modification is often performed under virtually anhydrous conditions and at temperatures of up to 70 degree, most processes still use mesophilic enzymes. Dear students, the next heading is the biocatalysis. Biocatalysis can be defined as the use of natural substances to speed up or catalyze chemical reactions. These natural substances can be one or more enzymes or cells. An enzyme is simply a protein catalyst. Every reaction in any living organism proceeds in presence of enzymes. Enzymes help us to digest food, produce vital nutrients, move muscles, and do just about everything else you can think of. Enzymes are also used in your daily life to improve the performance of detergents, make beer and wine, process food, allow diagnostic laboratories to tell us what is wrong, and many other tasks that seem to happen automatically every day. Reactions carried out at high temperatures can be advantageous in cases in which the substrate or product has poor solubility at low temperatures and or the thermodynamics of the reaction causes higher yield at higher temperatures. In addition, biotransformations in the presence of organic solvents might lead to a change in enantioselectivity selectivity of the reaction, reversal of the thermodynamic equilibrium of hydrolysis reactions, and suppression of water-dependent side reactions. Thermozymes can be applied as biocatalysts in existing industrial processes to replace presently used, often polluting chemical reagents. This is especially true for the pulp and paper industry, which has problem with aggressive waste from the bleaching process. Thermo and hyperthermophilic enzymes have great potential for industrial use as their high resistance is not only to temperature but can also be towards chemical, organic solvents and extreme pH values at ambient temperatures. Dear students, let's discuss the last objective of today's lecture, that is survival mechanism of thermophiles. Number of studies have been conducted on the genome, transcriptome and proteome, as well as the widespread and accumulated multiple omics information concerning thermophiles, making it possible to systematically evaluate how these organisms survive under harsh thermal conditions. First is the genomic evolution of thermophile. Environmental changes such as temperature shifts induce genomic evolution, which in turn provides the bacteria with thermal tolerant abilities to survive under high temperatures. Such evolutionary changes could be achieved through horizontal gene transfer, gene mutations, gene loss. Gene mutation in thermophiles. Since DNA is more unstable at high temperatures, the DNA repair system should be more stringent in thermophiles to maintain genomic stability. A genomic analysis of mutations in thermophiles such as thermophilus revealed that base substitutions can occur at a low frequency in thermophiles than in mesophiles. Several of the identified genetic alterations contributed to thermal tolerance, such as mutations in the GLP-F and FAB-A genes, transforming the evolved thermotolerant strain with the wild-type allele of GLP-F reduced fitness under high temperatures, whereas the mutation in FAB-A predictably increased the degree of saturation in membrane lipids, which is known adaptation to elevated temperatures. Next, genomic reduction and gene loss. The genome sizes of thermophiles are generally smaller than those of non-thermophiles, since all the species that live at temperatures greater than 60 degrees have genomes smaller than 4 MB, whereas all species with genomes larger than 6 MB live at temperatures lower than 45 degrees. As expected, the protein lengths and the number of protein family members in thermophiles are reduced compared with their homologous counterparts in non-thermophiles due to the small size of the genome of thermophiles. Second is the proteomics, that includes more disulfide bonds in thermophilic protein. Using genomic calculations, it was found that some thermophiles have a higher number of disulfide bridges than do mesophiles, and it was proposed that certain thermophiles use disulfide bonding as a major mechanism for protein stabilization. Next is the stable and efficient protein synthetic machinery. Heat shock proteins are the best known proteins that respond to heat stress and protect against cellular damage induced by heat stress. The upregulation of the heat shock protein genes in thermophiles has been widely observed at both the transcriptional and proteomics levels. Next is the protein complexes. Proteomic analysis of the thermophiles demonstrated that the number of detectable proteins at higher temperatures is obviously lower than that at the optimal growth temperature, additionally indicating that the thermostable proteins are concentrated in certain functional categories that should be necessary for thermophile survival under high temperatures. Next, we have the transcriptomics. It includes base biases of thermophilic genomes. Genomic structure in thermophiles is thought to be more stable than that of the mesophiles. Although the content of guanine and cytosine in the genome are important indicators of DNA stability, large-scale genomic comparisons between the thermophiles and the mesophiles have been conducted to evaluate the nucleic acid compositional differences. The GC content in some thermophiles is different from that of mesophiles, such as thermophilus with a GC content of 69.41%, Geobacillus costophilus with 52.1%, and Thermus species strain with 68.6%. Students, that's all about today's program. Hope you have understood and enjoyed. Thank you and goodbye.